3 million crabs were unleashed into a marsh where nothing lived, a radical experiment on a scale rarely seen. Florida's most barren coastline became a battleground for science and survival, but no one saw what truly killed this ecosystem in the first place. Who, or what, destroyed a thriving wetland, and could an army of creatures revive it against all odds? The answers would challenge everything known about ecological restoration. A stretch of Florida shoreline, once alive with the crackle of marsh grass and the flicker of minnow schools, now lay silent and empty. The mud stretched for acres, gray and motionless, broken only by the wind and the faint shimmer of standing water. No birds called, no fish darted in the shallows, and the air hung heavy with the scent of decay. This was not a scene of natural ebb and flow, but a place stripped of its pulse, a casualty of ecological collapse. Into this silence, a single act of audacity unfolded. At the direction of the Florida Coastal Restoration Initiative, trucks rumbled to the edge of the marsh, each one carrying crates packed with life. Workers in rubber boots moved with practiced urgency, their movements rehearsed and methodical. They carried plastic bins heavy with crabs, thousands upon thousands, each a tiny engine of change. There was no ceremony, no crowd, only the steady rhythm of release. Lids came off, crates tipped, and the mud began to move. 3 million fiddler crabs, all at once, scattered into the lifeless marsh. The ground, still moments before, erupted in a living wave. Claws flashed in the sunlight, legs churned the mud, and burrows appeared as if conjured from beneath. It was a spectacle of scale rarely attempted in ecological history, a deliberate flood of organisms into a place where nothing else had survived. Before this moment, the marsh had lost its identity as an ecosystem. Salt marshes are more than just grass and mud. They are engines of productivity, filtering water, storing carbon, and sheltering the young of countless species. Without roots to anchor the soil, the marsh had slumped into flat, exposed mudflat, stripped of the dense grass that once held it together. Fish nurseries vanished. Bird migrations bypassed the area. The loss rippled outward, touching fisheries, storm buffers, and water quality far beyond the shoreline. The scale of the release was staggering. 3 million crabs in a single coordinated operation, covering acres of barren ground. The logistics alone pushed the limits of restoration science. Every crab had been counted, transported, and selected for survival. The Florida Coastal Restoration Initiative, a coalition of state agencies and research partners, had gambled on a single dramatic intervention. The hope was simple, that millions of burrowing crabs could restart the engine of life, stirring oxygen into the mud, breaking up toxic layers, and making space for the roots of new grass to take hold. But the marsh was not just empty, it was wounded. Years of neglect, pollution, and rising seas had stripped away every sign of resilience. The question now was whether the movement of millions of crabs could do what science alone could not: bring a dead landscape back from the brink. As the last crate emptied and the workers stepped back, the marsh began to shift. The silence was broken, replaced by the faint, persistent rustle of claws on mud. For the first time in years, the ground was alive again. The outcome and the true cost of this gamble would take time to reveal. Rumors and speculation swirled in the wake of the crab release, but beneath the spectacle lay a simple, urgent question: what had gutted the marsh in the first place? The answers, as local field reports began to show, were tangled and far from obvious. Community liaisons, trained to read the subtle signs of marsh health, returned with notebooks full of clues. In one sector, water samples revealed nitrate and phosphate levels several times higher than historic averages. The source traced upstream, fertilizer runoff from distant fields and leaking septic tanks, feeding a silent bloom of algae. The algae choked the water, sapped oxygen, and piled up in thick mats, smothering the roots of marsh grass. But nutrients alone did not explain the scale of collapse. Hydrology maps from the previous decade told another story. Satellite data showed that tidal flow had been pinched off by a network of aging dikes and culverts, built decades earlier to control mosquitoes and reclaim farmland. The result was stagnant pools where salt water should have pulsed in and out, flushing toxins and keeping the marsh in motion. Without the tides, the soil grew sour and compacted, trapping sulfides and starving the roots of oxygen. Sea level records added a third layer to the puzzle. Over the last 20 years, the local gauge at Cedar Key registered a steady rise, almost 9 centimeters, outpacing the marsh's ability to build new soil. The roots of cordgrass, once thick enough to hold the mud together, now dangled in the water. Each storm peeled away more sediment, flattening the marsh and leaving it open to the next high tide. Field teams uncovered one more blow, the tracks and burrows of invasive herbivores. Nutria, imported decades ago for fur, had multiplied out of control. Their feeding habits left wide scars, chewing through young shoots and tunneling into the banks. In some transects, nearly half the remaining plant cover showed signs of grazing. The combined effects of these 4 drivers, nutrient overload, blocked tides, rising seas, and invasive grazers, turned a living marsh into a bare, sinking plain. Each factor left its own fingerprint, but together they stripped the land of its resilience. For the restoration team, the challenge was not just reviving life, but untangling the web of forces that had killed the marsh in the first place. Beneath the surface of the marsh, an architecture began to take shape. Each fiddler crab, no larger than a thumb, set to work with a singular purpose: digging. Their claws, evolved for both display and excavation, carved narrow tunnels into the dense, compacted mud. In just hours, the flat, airless plain transformed into a honeycomb of burrows, some plunging as deep as 30 centimeters. These tunnels were more than shelter. They acted as lifelines, channeling oxygen into soil that had suffocated for years. Oxygen diffusion in the marsh had nearly stalled before the arrival of the crabs. Without plant roots or burrowing animals, the upper mud layer sealed over like a crust, trapping toxic gases and stifling the microbial communities that drive decomposition and nutrient cycling. As millions of crabs churned the soil, oxygen seeped down through the walls of their burrows, breaking up the anoxic barrier. The chemistry of the mud began to shift. Sulfides, once trapped and toxic, started to dissipate, while beneficial bacteria found new footholds along the burrow linings. Each tunnel created a microclimate, a pocket of possibility where life could take hold. The physical changes were immediate and measurable. Soil bulk density, a metric for how tightly mud particles are packed, dropped by nearly a third in the first 2 weeks. Looser soil allowed water to drain more freely, carrying away excess salts and making space for the roots of pioneer plants. In restoration trials elsewhere, similar increases in burrow density have been linked to a 15 to 20% rise in root penetration depth within a single growing season. Plant growth, once stunted by hardpan mud, accelerated as roots chased the oxygen-rich corridors left by the crabs. The burrow geometry itself played a role in the transformation. Some crab tunnels branched horizontally, connecting neighboring burrows into networks that spanned meters just below the surface. These networks acted like tiny aqueducts, moving water and nutrients laterally through the marsh. In sections where burrow density topped 200 per square meter, soil scientists recorded a doubling of microbial respiration rates, a sign that the base of the food web was stirring back to life. For the restoration team, these changes were the first proof that the gamble might pay off. The lead ecologist, notebook in hand, measured burrow openings and collected soil cores, watching as the numbers shifted day by day. Each metric, including oxygen penetration, bulk density, and microbial activity, told a version of the same story. The crabs were not just surviving, they were engineering the marsh from the inside out. The ground, once inert and resistant, now yielded to the combined force of millions of tiny architects. The stage was set for a new chain of biological responses, as seeds, microbes, and invertebrates began to sense opportunity in the freshly aerated mud. At dawn on the first day, the restoration team fanned out across the marsh, carrying clipboards and portable sensors. They moved quietly, boots sinking into the loosened mud, eyes scanning for signs that the millions of crabs released just hours before were more than a spectacle. The team's main tools were simple, handheld oxygen meters, soil probes, and a network of wireless sensors staked into the ground before the release. These sensors, linked by a low-power mesh network, began to transmit the earliest data, numbers that would reveal whether the experiment was taking hold or vanishing into silence. Within 24 hours, a new pattern emerged on the digital burrow density maps. Where the mud had been unbroken, clusters of dark pinpricks now dotted the grid, each one a crab burrow counted and mapped by the field technicians. In the densest patches, the burrow count soared past 200 per square meter, a density rivaling the healthiest marshes along the Atlantic coast. The field team tracked these changes in real time, updating maps on tablets as they moved from plot to plot. The transformation was not just visible on the ground, it was measurable, pixel by pixel, on the screens in the mobile lab. Oxygen readings told a second story. Before the release, the marsh's upper soil layers had registered barely a trace, less than 0.5 milligrams of dissolved oxygen per liter, a level inhospitable to most life. By the end of the first day, sensors placed inside and around active burrow clusters recorded a sharp increase, some spiking to over 4 milligrams per liter. This surge was not uniform. In areas where crab activity was thickest, oxygen levels climbed fastest, while in pockets where crabs had yet to settle, the mud remained stagnant. The technicians compared notes, marking zones of rapid change with colored flags, their excitement growing with each new data point. Invertebrate counts, gathered by hand-sieving soil cores, added another layer to the picture. The marsh had been nearly sterile, but now, within 72 hours, the first wave of opportunists began to appear. Tiny polycate worms, nearly invisible to the naked eye, wriggled through the loosened mud, followed by early colonists like amphipods and juvenile snails. Field logs recorded each new arrival, noting both the species and their abundance. These numbers, though small, hinted at a chain reaction. Burrowed mud meant more oxygen, and more oxygen meant the return of the creatures that formed the base of the marsh food web. The field technicians, some with decades of experience, swapped stories at the end of each shift. One described how, in a single transect, the soil had changed texture overnight, from dense, sour clay to a crumbly, almost sweet-smelling mix. Another recalled the sight of a blue heron landing for the first time in months, drawn by the movement in the mud. The data was clear, but the early signs of life were just as powerful in the field. The faint hum of insects, the glint of minnow fry darting in the shallows, the first footprints of raccoons along the water's edge. Mapping tools layered these observations into a living record. By the end of the first week, the project's dashboard showed a patchwork of change: burrow density maps, oxygen heat maps, and species checklists growing more complex with each passing day. The marsh, once a blank slate, now pulsed with the first signals of recovery. The restoration team, buoyed by the numbers and the sights, prepared for the longer timeline ahead, knowing that the hardest work of rebuilding an ecosystem was just beginning. Official records from the county office paint a troubling picture. The marsh, once protected by a patchwork of overlapping policies, slipped through the cracks as regulatory attention shifted elsewhere. Permit logs from the last decade show a series of exemptions granted for water management projects. Culvert upgrades, mosquito abatement dikes, and shoreline hardening, each one stamped with language that prioritized public safety over ecological continuity. Enforcement files, thin and incomplete, reveal how routine inspections fell off after funding cuts in 2012. The state's Wetland Oversight Committee, responsible for monitoring cumulative impacts, never required a comprehensive review for the stretch of coastline. Instead, piecemeal permits were issued by separate agencies, each one focused on a narrow slice of the problem. Community liaisons recall meetings where concerns about tidal flow and nutrient runoff were raised but rarely addressed. The official minutes from a 2014 township hearing record, a resident's question about marsh die-off, answered with reassurances that existing permits covered all necessary protections. In reality, no single agency had mapped the full extent of hydrological changes or tracked the slow buildup of agricultural runoff. The permit gap here was not a clerical error, but a structural blind spot. No one was tasked with seeing the whole system unravel. As the marsh faded, responsibility dissolved into a haze of overlapping jurisdictions and unsigned forms. Climate models now project that extreme weather events, like hurricanes, heat waves, and sudden floods, will strike with greater frequency and force along the Gulf Coast. For the revived marsh, every storm season brings a new test. Even as the burrow networks multiply and the first grasses return, the ground remains vulnerable to saltwater surges and erosion, which can undo years of recovery in a matter of days. Scientists warn that this experiment, while promising, rests on a razor's edge. The crab's engineering may buy time, but it cannot halt the rising tide or filter out the next wave of nutrient runoff from upstream fields. Attempts to replicate this strategy elsewhere have met mixed results. In northern marshes, where fiddler crabs have migrated as waters warm, their burrowing has sometimes weakened plant roots instead of strengthening them. Cordgrass in these colder regions lacks the evolutionary partnership found in the south, leading to a 40% drop in root biomass and leaving the soil more exposed. The Florida case stands apart, shaped by unique local conditions, soils, tides, and a history of plant-crab coexistence that cannot be easily exported. Restoration scientists now face a difficult reality. While the spectacle of millions of crabs reviving a dead marsh offers hope, it is not a universal fix. Each site demands its own solution, tuned to the threats that linger on the horizon. The future of these marshes depends not just on bold interventions, but on vigilance, adaptation, and a willingness to confront the limits of what ecological engineering can achieve. Restoring a dead marsh isn't about spectacle. It's about understanding the intricate balance that sustains life. Today, salt marshes are vanishing at alarming rates, with over 50% lost globally. Each experiment, real or imagined, reminds us ecosystems heal only when we respect their complexity. The next intervention will matter, not for its scale, but for its humility. What would you risk to bring a landscape back to life?